All right, guys, here we go. Final chapter, chapter 24. We got carbohydrates, lipids, and protein metabolism. This is like where everything comes together. All right. So, digestion, we have like three basic veins we can go through: triacylglycerols, fats, hydrolyzed fatty acids, and glycerol. We talked about this. Our carbohydrates, hydrolyzed to monosaccharides, and proteins, hydrolyzed to amino acids. All of this is converted to acetyl CoA. It enters the citric acid cycle, and then the electron transport occurs. So just to make sure that we understand it, like here, we got this triacylglycerol. There's our, we have our glycerol compound here. There's our fatty acid change. We got our ester bonds here. Three waters, it separates glycerol, fatty acids. Carbohydrates, here's a disaccharide plus water, breaks apart. We get two glucose molecules. This is the alpha anomer, and there we go. There's our monosaccharide. And then proteins, valine, alanine, and glycine. I picked ones that were really easy to draw. These break apart. Again, we're using water. These bust apart into their amino acid components. So that's what we have. All right. So now we're going to go to the basics. All right. Carbohydrates, we have glycolysis converts glucose to pyruvate, which is then metabolized to acetyl CoA. There you go. All right. Lipids, this is fatty acids. Fatty acids are converted to thioesters, which oxidize stepwise procedure to form acetyl CoA. All right. There we go. In number 4, this is slide 4. Proteins, amino acids are formed from hydrolysis. That's how we break apart their bonds, are assembled into new proteins without any other modification. Or if they're not used for protein synthesis, then they're catabolized for energy, and the amino group is converted to urea, and we pee it out. All right. So here we go. Number 5. This is step 5. This is a biochemical reaction. Cells use the basic principles of organic chemistry. That's why we do that first. All right. Enzymes and coenzymes allow cells to carry out transformations that are difficult to replicate in a lab. So substrates, they have several functions. You guys can read this using products, reactants, enzymes, coenzymes, and other materials as possible to understand and analyze the biological process. Summation. This is super complicated stuff. What you're trying to do is study what your body does on a molecular level as you eat food. You can't just look at that. It becomes very hard. And so one of the questions I like to think about here is, how is this stuff studied? Like, what do you actually do in order to figure this out? And so it's pretty incredible that we know what we know at this point. All right. Um, slide 6, like you could see that there's not much going on here. We have fructose 6 phosphate. All right. Right here. Fructose 6 phosphate. We're going to use ATP, and that's going to become ADP. All right. There's a kinase enzyme, so we're transferring phosphate, and boom, we get fructose 1,6 biphosphate and ADP. So the AT or the phosphate group transferred from ATP to the fructose 6 phosphate. So here we go. We just did that. Phosphate added fructose 6 to form this. The phosphate comes from ATP. That makes energy, and so when ATP does that, you get a little burst of energy. The loss of phosphate, ATP becomes ADP. We're going to turn it back in a little bit. Kinase enzymes indicate that phosphate is transferred. Now, you're going to see these P's with circles around them. That's phosphate. That phosphate's a long word, and I want to have to write it all out. So start kind of getting used to that. Here we go. This is an enzyme catalyzed reaction. We have pyruvate here, super important from the citric acid cycle, decarboxylase. So we remove a CO2 and we add a hydrogen. No coenzyme is used because there's no reduction, oxidation, or thioester. We don't have to worry about that. Here, we have our phosphate group out here. So we have glyceraldehyde 3-phosphate dehydrogenase. That's a mouthful for an enzyme. But don't worry, again, we're not drawing it. We have this. We're going to use NAD plus, and we're going to take off 2 H's. So this is getting oxidized. All right. Here, boom, as we move over, that last carbon is oxidized. We have an oxygen added, plus we've removed some hydrogen. So there you go. Oxygen added, hydrogen removed, and oxidation NAD plus is the oxidizing agent. There's a whole lot going on. All right.